Hello, everyone, and welcome to the third week of the SciAm Summer Colloquium series. I'm very pleased to introduce Pete. He is in his third year of the Computational Science PhD program. His advisors are Naveen Vadia and Dominic Wadars, and his research is developing mathematical models of cystic fibrosis lung infections, and can't wait to hear all about it today. Take it away.
All right, hey there. You turn on screen share, Abe?
Yeah, one sec. Sorry, I forgot about that. Okay, should be good to go.
Still not yet.
Okay, so you should be the host, so you should have permission to do it. There you go.
There we go. Excuse me. Let me get my pointer going. All right, so hey everybody, I am Pete Ewell. I've been working on this project for a couple years now with my advisor Naveen Vadia, and it is over mathematical models for cystic fibrosis infections. We're working on this in collaboration with some folks over at the VII, mostly Forrest Rower, and Greg. Hey there, glad you could make it. We're going to be working on this in the future too. So this is an outline for the talk today. I'm going to start going over some background. There's quite a bit of background to understand, to kind of get the idea of these models that I'm going to be talking about. Then I'm going to go through an ordinary differential equation model of the bacterial populations in these lung infections. And then I'm going to move into some agent-based models. Okay, so cystic fibrosis is a genetic condition. It's caused by this defect in a gene called the CFTR. And that gene regulates, it's a chloride transport across cell membranes. And when it screws up, like in cystic fibrosis, it prevents water from being properly transported across cells. That can cause a lot of bodily fluids to become dehydrated, and as a result, they become very viscous and slow moving, and this causes a lot of problems in a lot of different parts of the body. People with CF, they have trouble gaining weight because it screws up with the stomach, and they aren't able to properly absorb food, absorb or digest food. In males, it can screw up the reproductive system. The thing CF is most known for is these lung infections that it causes. The buildup of fluid in the lungs is mucus. So ordinarily in lung airways, you have this thin layer of mucus going around the inside of the airway tubes. And that thin layer of mucus, together with organelles the epithelial cells have called cilia, kind of keep things moving through the lung airways so that nothing is able to really just stay put. And so any foreign pathogens, or any dust or anything that gets down there, it gets cleared out. In CF, this mucus is so thick that it can't really move, and things get clogged up. And also this mucus, it also has a lot of nutrients in it that bacteria can come in and reproduce using that. Okay, so what my research is in is modeling the bacterial populations that move into this clogged up mucus. And in particular, I'm going to be looking at the effects oxygen has on it, because a lot of these bacteria are aerobic, which means they use oxygen for energy production. But some of them, in parts of the mucus, it becomes harder for oxygen to penetrate deep into this clogged up mucus. And so parts of the mucus can become hypoxic. So there's no oxygen getting there, and that makes room for anaerobic bacteria to start colonizing there. These anaerobic bacteria are problematic because they use another energy source. They use another method of energy production, which is fermentation. And that fermentation has a lot of toxic byproducts that is what actually makes people feel sick a lot of the time when they have these lung infections. So we want to understand the exact role oxygen plays in the dynamics of these bacteria. So this approach kind of started with Doug Conrad and Forrest Rower in the VII. And basically they're hypothesizing that we can categorize these bacteria into two broad communities. There's one called the climax community, which is generally aerobic. The main player here is called Pseudomonas aeruginosa, which is one of the most common pathogens found in CF lung infections. That's the thing that clinicians target most often when people go to the hospital with these lung infections. And there are also other aerobic bacteria in this climax community. The attack community contains aerobes or anaerobes, so oxygen is going to be toxic to most of those. It does also have some what are called facultative anaerobes, which are bacteria that can use oxygen if oxygen is available, but if oxygen is not available, they'll switch to fermentation. So they can also be problematic if there's low oxygen. Okay, so the important thing here is that the attack community is the bad one. We want to try to keep the attack community low because if there's lots of attack bacteria, then that's going to cause what are known as exacerbation events, what I'm going to come to next. However, both of these bacteria populations are pathogenic. So the climax community over decades will also cause severe disease and death eventually, but the attack community is more acute. So if you have more attack bacteria, you're going to be sick faster. Okay, so these periods of severe disease are known as pulmonary exacerbations, or CFPEs. These are the leading cause of death in cystic fibrosis patients. Again, the main idea of this research is that we're hypothesizing that these attack bacteria are the main cause of these CFPEs. So we want to try to keep them low. Clinically, what they tend to do is just try to kill everything with antibiotics. In the last couple of years, there's kind of been better understanding of what antibiotics or of what bacteria are actually causing the more serious disease. So there's been developments in targeting these more pathogenic attack bacteria. But historically, I think they've just used a lot of broad spectrum antibiotics because mostly it's the Pseudomonas that is the main CF pathogen. Since we're looking at oxygen, though, in our population models, though, we also want to see if we can try to keep the attack community low, possibly using oxygen. So that's what we're going to talk about a little bit later, too. So now I'm going to start going into what I'm doing with our model, with my modeling. So in the next slide, I'm going to show ordinary differential equations of these models. And they're going to be based on logistic growth. So we're going to have some growth rate for each of the bacteria populations. These growth rates are going to be what are called Emax functions or Hill functions. And what they are saturating functions of oxygen. So this graph here is a graph of what the growth rate will be for a certain amount of oxygen. So oxygen is increasing along here along the x-axis. So the blue curve is going to be the growth rate for the climax community. So if you have low oxygen, the climax community is going to have a low growth rate. As you increase oxygen, it's going to have a higher growth rate. The attack community growth rate is going to be reversed. So the attack growth rate, if there's no oxygen, that's when it's going to be the highest. As you increase oxygen, its growth rate is going to drop off. So I'm going to show these parameters in the next slide, but the three main parameters for these Hill functions are the maximum growth rate. So we call this beta. This beta is the maximum possible growth rate. So if you look at this red curve and oxygen at 0, this is the value for beta in this particular example. B is the half saturation constant. That's going to be a concentration of oxygen, and it's going to be the concentration of oxygen that gives you exactly half of your maximum growth rate. So looking at this blue curve, B is going to be somewhere around here because that's where your growth rate is exactly half of its maximum. And then there's also the slope factor N, which determines how steep you go from high to low. So if N is a very large number, this curve is going to be very steep. And for very large N, this actually this curve is actually going to be close to a binary function. Okay, so here is the ODE model. We're going to have population or equations for the climax population, the attack population, and the amount of oxygen that's present. So these equations are just logistic growth for the two populations, where there's some carrying capacity K, which in this model is just sort of meant to represent the total available space that these bacteria have to grow in. So as the total population approaches K, both growth rates are going to drop off and they're not going to be able to grow anymore. And then they're going to have some intrinsic death rate or possibly death due to antibiotics in these DC and DF terms. We're going to unpack those quite a bit coming up here. But then also, because oxygen is toxic to these attack bacteria, there's also going to be a killing term of attack bacteria due to oxygen. So when the attack bacteria interact with oxygen, they're killed at this rate Q. Oxygen is flowing in at a certain rate, is decaying at a certain rate, and it's also being consumed by the climax bacteria because they're aerobic, they consume the oxygen, so that's going to lower the amount of oxygen available. So this is a cartoon of these dynamics. Again, we have a certain amount of space to grow in. We have the two bacteria populations being born at certain rates and dying at certain rates. The attack population is also killed at this rate Q due to oxygen. Oxygen is flowing in, being consumed by C, and it makes C grow and F grow slower. And then these growth rates are defined by these hill functions here. So again, if you have a very large amount of oxygen, this term is going to approach beta. Because the attack or the climax bacteria also contains some facultative anaerobes, they can actually also grow if there's still low oxygen. So we just include this plus R term, which doesn't depend on oxygen, just to be the most correct. But this R term doesn't have that big of an effect on the dynamics. And then the attack growth rate is just going to be the reverse hill function of the climax. So here, if you have very large oxygen, this term is going to be close to 1, so this term is going to approach 0. Okay, we're going to fit this model to some data that we have from the UCSD cystic fibrosis clinic. So this patient was undergoing an exacerbation event. Over about 40 days, they had sputum samples collected, and they went through different regimens of broad spectrum and targeted antibiotics. So initially, this patient was being treated with broad spectrum antibiotics, which mostly killed all the bacteria. After a while, they got a little bit better, so they were released from the hospital and stopped taking those antibiotics. But what happened then was these the attack bacteria rebounded. That caused them to get sick again very quickly. So they went back to the hospital. This time they were prescribed antibiotics that targeted the attack bacteria. Okay, so now I'm gonna just unpack these antibiotic treatments into the death terms that I use in the fitting. So like I said, Pseudomonas is the most well-known pathogen. So that is what is most often treated with and what is in mind when they prescribe antibiotics. So these are some antibiotics here that were used during this broad spectrum period to try to kill all the bacteria. In the latter part of the data, when the targeted antibiotics are used, that's an antibiotic called clindamycin, which is known to kill streptococcoids, staphylococcoids, and anaerobes, which are bacteria that we have in the attack population. So going back briefly to those death terms, so the DC and the DF, when we do this, when we incorporate these various antibiotic treatments, we're going to have DC is DN plus DBS, where DN is just the natural death rate, so both populations will have that in common. And then DBS is going to be the death due to broad spectrum antibiotics. So in period A, this DBS term is going to be positive. Since the broad spectrum antibiotics don't kill the attack bacteria as much as the climax, we still have this DBS, but we have this reduction factor gamma, where gamma is just 0, 1. So that the attack bacteria are not killed quite as much by the broad spectrum antibiotics. In what we call period B, when the patient's released, they're off all the antibiotics, so both of these terms are going to be 0. And then when they return to the hospital in period C for the target antibiotics, there we have DBS equal to 0, and epsilon, which is the killing term due to clindamycin, is going to be positive. Okay, so this is the ODE model fit to the data set from that patient. In period A here, we see that the populations are pretty stable. If you continued this, you would see that these populations will continue at kind of a equilibrium there. And the increased death due to the broad spectrum antibiotics kind of is suppressing the attack community more so than the climax bacteria. That may also be due to initial conditions, but the thing to notice here is that when these broad spectrum antibiotics are in use, the climax community is able to suppress the attack bacteria, and after a certain amount of time, the person was feeling better and feeling well enough to end the treatment regimen and go back home. When they did that in period B, so period B, this middle panel here, all the Vanix are off, and when that happens, the attack community is no longer being suppressed, and they come up here and are able to overtake the attack population or the climax population, and this causes the worsening of the disease. At that point, again, like I said, they go back to the hospital, and this clindamycin kills the attack bacteria only, and so that's why you see this drop off here. So that is kind of the narrative that we've kind of attached to this set of data, and the ODEs fit to it. Okay, so we want to analyze this ODE model a little bit. And I do this in 2 ways. First, we're going to look at the long-term dynamics. So that's just going to be the possible long-term outcomes for these bacteria. And then after this, I'm going to talk about transient dynamics. And we're going to want to see how long it takes for these exacerbation events to occur. Okay, so this is a little bit of math here. I'm not going to say too much about it, other than I'm going to take oxygen to be in quasi-steady state. So set the oxygen equation equal to 0 in our model. We do that, we can just write this as a two-dimensional ODE system, which means we can do phase plane analysis. So the equilibrium of this reduced model is just going to be where the null lines intersect. So that'll be where C prime equals F prime equals 0. That'll give you a set of lines in the plane, and where those lines intersect is going to be the equilibrium values of this model. So this model is going to have 4 biologically relevant steady states or equilibria. There's going to be an extinction steady state at the origin where both populations go extinct, 2 competitive exclusion equilibria where one population goes extinct and the other survives, and then 1 equilibrium of stable coexistence. Okay, so this is just a plot of these null lines in the C and F plane. So there's an equilibrium at the origin here that I've left off. And then this blue line is just this C prime equation set equal to 0. And red line, same thing with the F equation. So the equilibrium are going to be at this point where they intercept, and also where each of these lines intercept with the other axis. So this red line hits the F axis here. So this is going to be the equilibrium value of the F only equilibrium. Same thing with the C. It intersects the C axis down here. So that's going to be the C only equilibrium. When I look at the stability, I didn't put much on the slides on this, but when I look at the stability of each of these equilibrium points by doing linearizing and then eigenvalues, I found that as long as this equilibrium point is in the positive sector or the first quadrant, it's going to be stable, and it's the only stable equilibrium point. So if this intersection point is located in the first quadrant, it's going to be the only stable equilibrium of the model. These 2 competitive exclusion points are going to be unstable, and the origin is going to be unstable here as well. So we're just going to have a node at this point. When I do bifurcation analysis, which is what the next couple of slides are going to be on, we're going to see that this equilibrium point will move throughout the CF plane as we vary parameters. And then, so we're going to have transcritical bifurcations if this point moves such that it crosses the f-axis or such that it crosses the c-axis. So if this equilibrium point moves such that it crosses this axis, there's going to be an exchange of stability. The coexistence equilibrium will no longer be biologically relevant for one thing, but it will also become unstable mathematically, and this F-only equilibrium will gain that stability. Okay, so one thing I haven't mentioned so far is that we kind of define mathematically as an exacerbation event to be when the F population is over 50% abundance. So in this point, if you drew a straight line here, we would see that at equilibrium, F is over 50% abundance. So this would be an exacerbated state. So that's what we're going to be looking at a little bit in the next couple of slides. But so first I'm going to talk a little bit about this death rate, because that's kind of the most interesting parameter we can mess with here in terms of treatment options. So as I said, if we vary a parameter, in this case, we're going to be doing it with D. And D is just going to be a common death rate in all this analysis. So if we don't have, in the absence of antibiotics, we just assume that these bacteria all die at the same rate. So if we trace out a range of D, then the equilibrium point is going to move throughout the CF plane in this manner here. So if we have very high death or very low death rate, then that's going to favor the climax bacteria. So which is good because that's going to avoid an exacerbated state. As we increase this death rate, we see this kind of maximum area where this is going to be the point that's most favorable for the attack bacteria. So clinically, that's what we want to avoid. And this is interesting because there definitely is this kind of optimal point where if we keep increasing the death rate, which would be using either more or more powerful antibiotics, the attack population is going to drop off again at equilibrium. And eventually, if we just totally nuke everything with very strong antibiotics or some other kind of treatment, then that is going to eventually make the origin stable and where both populations go extinct, which probably isn't very realistic or even desirable. So this is interesting. This tells us that there's a kind of a sweet spot of antibiotic treatment where the attack population is the best off. These two figures are just some more stability analysis or not stability exactly, but we're looking at the level of F. So we're interested in seeing how prominent the attack bacteria are at equilibrium based on a couple of different parameters. So over here, I'm going to start with this right-hand figure. This is that half saturation rate or concentration, the B parameter, and then this is the slope factor. And then this contour diagram here is just showing the areas where F is better off. So if we have high B, high F, then F is going to be more prominent at equilibrium, and we want to avoid that. And on the other hand, if we have low B and or low F or low N, then that's going to keep the attack population low at equilibrium. The figure on the left is the same, just with a couple of different parameters. In this case, we have the oxygen toxicity rate. So clearly that is going to be, high Q is going to be bad for F. And also the consumption rate, which, the consumption rate is also kind of interesting because F likes having a high consumption rate of oxygen, because a high consumption rate means that oxygen is being consumed faster, which means less of it is around to kill attack bacteria. So see, F then clearly favors having a low toxicity rate and a high consumption rate. Okay, so moving on to, so that's all I have for the steady state analysis. So now I'm going to talk about the transient dynamics and how quickly these exacerbation events occur. So this figure on the right is just kind of the base case, what we're looking at in terms of this exacerbation event. So if we're starting with some low amount of F, we're interested in seeing at what point F becomes more than 50% abundant. So this time is what we're interested in here. So this is about 6 days here for this set of parameters that we're running. So now I'm going to do the same thing with varying parameters and now seeing how long this switch in population takes to occur. So again, there's something interesting here with the death rate, which is that there's clearly this trough here where if you have an intermediate death rate, the switching can happen faster. Where if you have a moderately large death rate, then the switching takes longer, or past this point of about 5.2 something or so. If you increase the death rate past that, the switching never occurs. So at that point, the attack population is either going extinct or just staying at a level that won't ever reach 50% abundance. And then again, this is interesting. If there's kind of a very low, if there's not too high of a death rate, that also hurts the attack bacteria. So that's kind of an approach with antibiotics. But another thing, since we're looking at oxygen, it's kind of interesting to see what oxygen is going to do with these two. So this graph on the right is the same thing, but with our oxygen inflow rate lambda. So if you could think of this as either going on an oxygen mask, or maybe a hyperbaric chamber or something that just is going to increase the concentration of oxygen that you're inhaling. And again, but it's not surprising that if you are increasing the oxygen flow, then that's going to be unfavorable to the attack bacteria, which is good clinically. And again, just a couple more contour figures with the same, well, slightly different parameters. In this case, we have the death rate D and the oxygen inflow rate. So the same two parameters we were just looking at. But then in this case, we're just seeing how they behave together. So this big swath of blue is going to be a region or a parameter regime where the attack population is coming up very quickly. So that's bad clinically. And then for kind of a smallish range of these two parameters, we get this finite amount of time where it still happens, but it just takes a longer time. The switch happens, that is. And then in this white region, that's again going to be parameters where the attack population either goes extinct or is existing at a level that's below 50% abundance. Same thing with this figure on the right. Again, the two growth rate functions, so the slope factor and half saturation value. Again, the big blue area is going to be good for the attack bacteria and bad clinically. And we have this kind of narrowish sliver in the bottom left here where we're good, where either it takes a long time for the switch to occur or it doesn't occur at all. Okay, so that is kind of the story with the ODEs. And now we're going to change gears and start talking about what are known as these agent-based models. So the idea with the ODEs is that they are taking place in a well-mixed environment. So location doesn't matter in the ODEs. In reality, though, we know location does matter because bacteria, they reproduce locally and they consume oxygen locally. So if you're a bacteria and you're dividing, you're not going to place your offspring a long ways away. You're going to place it right next to you. And we can do that using these agent-based models. So what we're going to do here is first we're going to talk about a spatially homogeneous agent-based model, which has all the rules of an agent-based model, which I'll talk about in a second, but it is spatially independent. And the reason we do this is because this agent-based model is actually going to be a perfect analog of the differential equations. So we can validate the agent-based model this way using the differential equations. Once we've done that, then we can add in the spatial component and we can see what happens there. Okay, so this agent-based model is a stochastic process and it all just takes place in a grid. I'm saying it's a square grid, but it doesn't really have to be. It can be any kind of, it can be any shape domain. And this grid is full of spaces, and each space can either be empty, it can have an attack bacteria, or it can have a climax bacteria. Each bacteria has a certain probability to die, or a probability to die and a probability to reproduce. So what this simulation is going to do is going to say there's going to be a total number of bacteria in the grid. at the beginning of your time step. Each time step, you're going to randomly sample points in the grid until you've encountered n, that total number of bacteria you started with. When you hit a bacteria, you're going to follow some rules, and that's going to determine the bacteria's action at that time step. Okay, so this basically is just kind of the algorithm, you could call it, or the process, or the set of rules that these bacteria follow when they get selected. Or maybe it's more accurate to say that we're picking spots at random, and this is what we do based on what is in the spot that we pick. So at the beginning, we're just going to pick a spot randomly. If that spot is empty, we're going to go back and pick again because we can't do anything with that. If the spot's not empty, we're going to pick a number, and we're going to ask if that number is less than the probability to reproduce. If it is, then we're going to pick a new spot, and if that new spot is empty, then we're going to place an offspring cell there. If it's not empty, then we're going to do nothing. So that kind of gives the sequence of event for placing offspring. If we go back to the top, say yes, pick a space, yes, it's empty, pick a random number. It's not, this time it's not less than your probability to reproduce. We're going to ask, is it less than the probability to reproduce plus the probability to die? If it is, then we do nothing. If it's not, then we're going to die. So this framework just sets up a set of probabilistic rules for how these bacteria reproduce and die within this grid. And so now we should be coming up on the fun part, which is the movies. Okay, so this GIF is going to run on loop. And so if you look at this grid, the black spaces are empty, the yellow squares are attack bacteria, and the purple squares are climax bacteria. So you might want to wait for it to circle back to the beginning. I don't think I can pause it. So we might just have to go a couple of laps on it. Okay, so what we're doing in this agent-based model is we're setting the growth rates to be oxygen dependent in exactly the same manner as in the ODE. In the spatially homogeneous case, there is just a global amount of oxygen available. So that just means we have to update, we have an ODE governing oxygen that is updated each time step based on how many climax bacteria are in the grid. So this is a simulation of that patient data that I showed a couple of minutes ago. So initially, we're on broad spectrum antibiotics. That's keeping the population density very low, and it's keeping the attack bacteria relatively less abundant. When we go off the treatment, both populations, when we go off the treatment, the attack population shoots back up and the patient gets sick again. And then towards the end of the data set, we go on the clindamycin, and that comes in and nukes all the attack bacteria. So maybe go through this one more time so we kind of get the whole picture. So again, broad spectrum, killing everything. And then right about now, we're going to go off treatment. So everything's going to come back up, but mostly the attack. And then at the very end, we're going to go back on the attack only treatment, which is going to kill all the attack bacteria. Okay, so this is an ensemble of that agent-based simulation I ran with 95% confidence intervals. You see, the confidence intervals are very wide at the beginning because since the population density is a lot lower, that's introducing a lot more variability. So that's why we're seeing these wider intervals. That being said, this ensemble is not perfect. I need to do this a little bit more before it's ready for publication, but we are seeing generally pretty good agreement with the ODEs, which is the whole point of the spatially homogeneous model. Okay, so this is probably the most interesting part computationally of this project, which is going to be the spatially dependent agent-based model. So what we're doing now to introduce a spatial component is, first of all, when bacteria reproduce now they can only place their offspring in one of their 8 nearest neighbor cells. So that part's easy. Again, now if you were chosen to reproduce, you have to pick a spot at random from only the 8 nearest spaces. And if the spot you pick is empty, you reproduce there, and if it's not empty, you abort. So that part's pretty easy. The hard part is this local oxygen concentration. So what we have to do here is set up an ODE in each space of the grid. And this is what the ODE is for every space. So it's the same ODE k by k times. So this is a k by k ODE system that actually has to be solved for every step, for every time step of the agent-based simulation. So what we do algorithmically is we go through the whole grid, update the oxygen concentration, integrate the ODE system 1 time unit, and then we let the agent-based simulation run until it hits n individuals and they all reproduce or do what they will. And that is a full step of this agent-based simulation. So there's some new terms in this oxygen ODE. The inflow, outflow terms are the same as they are in the ODE. The consumption term is a little different. In this case, since we're just talking about individual bacteria in these spaces, I just call this C, yes, no. So the C, yes, no is going to be 1 if there is a climax bacteria in a spot and 0 if there's not. So it'd also be 0 if there's an attack bacteria there. G, which these two terms have in common, is just a diffusion rate. The lowercase m is the number of neighbor spaces a particular space has. So in most cases, you're going to have 8. But if you're on the boundary, you're going to have 5. And if you're in a corner, you're going to have 3. And then X total is the total amount of oxygen in all of your neighboring cells. So how these diffusion terms work is that however much oxygen you have is X, and you're sending M copies of that out into your neighboring spaces. On the other hand, though, your neighboring spaces have a total amount of oxygen, and some of that is flowing into your space each time step. So that's kind of how these diffusion terms work. And so now here the coup de grace is this simulation of the spatially dependent model. The thing that I notice here is that, well, so let me back up. So on the left, we have the bacteria. So that's exactly the same as the spatially homogeneous case. So purple are climax, yellow are attack, and black is empty. Then on the right here, this heat map is the amount of oxygen that's in a particular space. So what I think happens here in the spatially dependent agent-based model is that the attack bacteria need the climax bacteria to come in and consume oxygen. Otherwise, the oxygen kills them. So when you watch the bacteria going, they tend to want to follow around the climax bacteria, because if they're off on their own, oxygen is going to come in and kill them. So the attack bacteria, they really depend on the climax bacteria in this way. And so I think this is what they're going to be excited about the VII, because it's creating an ecological niche where the climax is altering the environment, and that's letting the attack move in. So I'll let this go for a little longer, and then I think I'll wrap up with conclusions. So there was one down here. So again, these yellow spaces, they want to clump together and they want to be near purple. If you get any, like down here where you get some yellow off by themselves, they tend to go extinct like that because oxygen is flowing in there and it's increasing their death rate. Okay, so it just did a lap there. Okay, so I'm going to wrap up. So I think I finally got around to finding some interesting therapeutic things that we can do with this model. It looks like, in particular, there's a certain range of antibiotic treatment where it benefits the attack, which you don't want, because if you have just a moderate amount of antibiotic being used, it's not going to be good if that lets a more pathogenic bacteria move in. So we want to avoid that. And then also we can kind of hold off on the exacerbation events by just adding oxygen to the system. I think the next thing to do which is going to be kind of my next major project, is just going to be understanding this spatially dependent agent-based model. The simulation I showed there is kind of arbitrary, just kind of very early days with that. There's a lot of study that needs to go into exactly how these bacteria interact when the oxygen is driving the dynamics. So that's going to be probably my work for the next several months. And then eventually, I think the goal for this project was possibly to introduce some phage therapies. So we'll see if we can cook something up over at the VII about that. That might be exciting. And some acknowledgements here. Cynthia Silveira was a postdoc in Forest Lab. She did all the sequencing for that data set, which I understand was a lot of work. And then Douglas Conrad is the pulmonologist that was responsible for that patient. And then Forrest and Dominic Wadars is my advisor at UCI, who kind of tutored me in how to develop these agent-based models. So that is all I have, and I can take your questions now.
Hey, Pete.
Hey there, Greg.
That is a really cool system. Let me just turn on my video real quick. Though at Telluride, not to disappoint you, we pretty much just went through and threw away the climax and attack model, and now we've added some more nuance, and now it's 4 distinct communities which have functional definitions which can sometimes overlap between the the more traditional categories. So
I had a feeling something like that might have been coming down the works.
Yeah. Currently, I'm sort of trapped in Colorado because of the heat wave. My car can't make it through the desert. But I should be back like on Monday if you want to come in, and I'll share my notes with you about everything that we talked about.
Yeah, sounds good. I kind of figured once you guys got back, I'd start going in once or twice a week.
Would there be any way to include eukaryotic cells in that in the intervening space, or in that agent-based model? Would we be able to sort of include lung epithelial cells as oxygen, like things that can draw down oxygen and then like may die because of proximity to an attack bacteria?
Yeah, and that's actually something I'd been thinking about long term with regards to like the scarring that happens, right? Yeah. Yeah, I don't see why not. Just take some work with the agent-based model, but like, yeah, mathematically that shouldn't be an issue.
I mean, that would be really cool, because I'm working on an in vitro model that should do that. We're going to try and sort of 3D print an airway with epithelial cells intact, and then introduce the bacteria and sort of watch them grow.
Well, and I'd actually want to tell you, let me go back to this. What you were telling me the other week about the mucus plugs is kind of what I had in mind when I was watching this, because it's not exactly like the anaerobes are inside, but like they want to latch on to the aerobic ones, because the aerobic ones are getting rid of oxygen. So that's kind of what this made me think of when I was working on this last night.
Yeah, the niche building, I think, is really important. And we think that oxygen drawdown rate, so when I look at this, I'm seeing a lot of empty space, but it would probably require most of that space to be filled with the aerobic community in order to draw down the oxygen enough for a true anaerobe to proliferate. But then we have those things like the microaerophiles, which are like strep. Strep is and staph, those are microaerophiles.
Yeah, well, you don't want to get hung up on the details of this particular one. Like this thing is basically a prototype right now, I would say.
No, I like this. This is a really cool tool, and I think it's heading in the right direction. I'm just thinking about how much more complex I would want to make it. I just want to give you a ton of work.
That's the name of the game, I think.
One more thing, Pete.
Yes.
You started with the background like mucus, but we never thought anything to think about the mucus, right? Do you have any feeling? Because your background is right away, if see a patient, there is a mucus, thick mucus. And when I clicked in my head, maybe we should, and maybe not this in first paper, maybe second thought, somewhere you should say or bring something mucus. How can we do that? Bringing whole mucus may be complicated stuff. It's a fluid dynamics, so many things there. But still, is there some straightforward way that we can bring mucus here?
I mean, I don't know. In this well-mixed model, like we're kind of considering taking like a patch of airway that's like basically empty except for oxygen fluid, and then like the bacteria are actually living in the mucus. So like definitely the ODEs, I don't think, are going to capture anything going on.
No, my point, first of all, we should know, maybe Greg can help in those kind of things. What does mucus do? Like both way. What's the role? If we know the role, then OD, we can extend that to maybe OD and PD combined model, separate background for the mucus or something like that. But I don't know right now. There is a mucus, I saw a picture, but actually what does mucus do or how is it formed? There is a role of bacteria for formation of mucus, or there is a role of mucus for the bacteria population, both ways. How much do we know actually?
Yeah, so Greg, don't like the anaerobes break down the mucus as a carbon source?
They do. And then the climax community that you're calling it here, like the traditional CF pathogens like Pseudomonas, when they are in anaerobic conditions, as long as they have access to nitrate available, they start producing alginate, which can be even more, what's the word I'm looking for? Recalcitrant than mucus when it comes to like mucociliary clearance. But are you talking about like what mucus does? Are we talking about what it does in like moving in 3D space, or are we talking about diffusion of nutrients or diffusion of oxygen, or what are we talking about? Or electron acceptors versus electron donors, or is it all of those?
We're talking about, because we don't know anything, so we're talking about anything you can tell us. So one thing you just said that if there is a mucus, if you have a diffusion of oxygen, if there is a mucus, then there is not a pure diffusion, right? There might be some subdiffusion or some kind of obstruction for the diffusion of oxygen. That one thing, having mucus environment or not mucus environment, maybe different dynamics you get. That one thing looks like.
So off the top of my head, having spoken to Robbie Quinn just like a couple days ago, was that mucus requires about 1.8 millimeters in order to reach full anaerobiosis, so like fully anaerobic condition. A bacterial biofilm can draw that oxygen down much more quickly, but in a mixed population of bacteria, so non-biofilm, you could end up with a lot less oxygen drawdown. And so like the actual depth of mucus required to reach like an anaerobic condition is something that we're kind of working on right now, trying to figure that out. And you may need epithelial cells in order to reach that gradient of oxygen.
Okay.
Although when you get to the smaller airway passages, then I think the mucus can be even more involved in impacting the diffusion of the oxygen. So that as the mucus builds up, you get more of those anaerobic areas developing. right? And you even can get mucus plugs where it completely plugs an airway passage and there's no oxygen passing through it, right? So, yeah, it's, and that was exactly what I was thinking, Naveen, what you had mentioned. I was wondering if, Pete, if there's any easy way, or it seems like it would be productive to try to build in a little more of that airway passage structure in your spatial model, right? Because not everything is adjacent to everything else. They're in airways, so there's sort of a restricted environment around them. So something to think about. I don't know, with it, you're getting some very nice and interesting results now, and it may be more complication than it's worth, but
yeah,
just to consider.
yeah, but overall your results actually looks cool, especially with the movie. And also what I like, even the homogeneous part, heterogeneous is you are still doing that. Even homogeneous part, actually, it is so much consistent with the exactly what you have with OD. At least one patient is a very limited patient that can be totally outlier. We don't know, but it's still, you have so much of thing. And when, by the way, when you draw that figure, you can draw the real time scale instead of 1200 something time scale. Draw actual how many days exactly.
Right.
But in your movie, you clearly show that okay, after this many days, there were more of this, and other one comes, then again it disappears. So it looks actually good. It's mimicking something. So that means this is a nice experimental silico experimental setting. So you can do a lot of experiment on that now, actually, parallelly.
Yeah, there is a couple issues I need to hammer out that I'll talk to you about probably next week.
Yeah. So, but definitely, like Ben mentioned, these are a little bit to make it more reality. That's how we can make the domain is like that, or mucus things and those kind of things. This will be really cool stuff, actually. So that's the next step we have to think of, that how we can make it more real kind of thing.
All right. Thank you, everybody, for the ideas.
It's good. Then I'll see you.
All right.
Thank you.
Yeah.
That was really
Come to
nice.
the lab and grow some bugs with me, and then we'll see how much realism, right, we can sort of stuff in there.
Right.
All right. Sounds good. All right. I'll talk to you two next week.
Okay.
All right.
Okay. Thank you. Bye.